Hello,
Hi, is this Donna McDonald McGinn?
it is.
Hi, my name is Johnny Sepp. Welcome to the 22Q Ironman podcast to raise funding and awareness for the charity. How are you?
Good. Are we live?
We are live on the air.
Okay.
Is that okay?
Well, this is a cell phone. It's not my office phone, and sometimes I get cut off on this phone, but we can certainly give it a shot. How are you?
I'm doing very well. Thank you very much. I'm a little bit worse for wear. We are in our 37th hour of broadcasting for 22Q awareness.
Okay,
But
for you, have you had a lot of protein bars?
um, I've had all of them. All of the ones I bought are gone, and I have. My father was nice enough he's on his way to bring me more. So I just want to give a quick.
Listen, we really appreciate you doing this. The entire 22Q community is absolutely thrilled, and we can't thank you enough.
Oh well, it's the least we can do. As you know, Joe Cello is a great friend of our show, and it seemed like a no-brainer when the idea was put out there, because I've done tons of reading and research, and it's really a cause and a foundation that I support fully, and think that more needs to be done to not only spread the word, but make life easier for those affected and so forth. So it's absolutely my pleasure. For everybody listening, I'm sorry, should I call you Donna or should I call you?
Sure.
Okay.
Donna is perfect.
All right, awesome.
I answer to everything, including "Hey, you."
Well, Donna is the recipient of the 2012 Angelo DeGeorge Memorial Medal, which recognizes an outstanding contributor to the understanding and treatment of the 22q11.2 deletion syndrome. The award was inaugurated in 2010 to commemorate the life's work of Dr. Angelo DeGeorge, and you're only the second recipient to receive that medal. That sounds like a big deal to me, and congratulations on that accolade.
Well, thank you so much. And, I really have to say that I was incredibly honored because Angelo DeGeorge was a pioneer in this field, and he had passed away in 2009, and that was the reason that the award was inaugurated in 2010 at our biennial meeting, which is why there have only been 2 recipients so far. And what I said at the meeting in Florida where I received the award is that unlike Peter Scambler, who was the recipient in 2010, the inaugural recipient, who's an MD who has done pioneering work himself in the area of detection, and he does really eloquent work in the mouse to try and understand what this deletion does to the heart and craniofacial features. And he runs marathons for the family support group in the UK, Max Appeal. And so I said, I'm not any of those things. I'm actually a person who has benefited from working at the Children's Hospital in Philadelphia, which is considered one of the 4 front leaders in pediatric healthcare and surgery. And so I've benefited from all the people who work here at CHOP, as well as all the relationships I've had with families around the globe, including the 22Q Foundation. So, I was the recipient, but really I receive it on behalf of all those people who helped me along the road for 20 years and who've inspired me to work with these families. And Joe and Paula, so Julia, their daughter with the deletion, who's just inspirational herself, and her sister Samantha, who's there for her, and the grandparents, Nona and Noni, and all of them, and it's just a wonderful thing. But thank you, I appreciate it.
Oh, you're very welcome, and they are a wonderful family. You had mentioned the Children's Hospital of Philadelphia. You're the Associate Director of Clinical Genetics at the Children's Hospital of Philadelphia. For those who might not know, what exactly does that entail?
So in clinical genetics, we see patients essentially with birth defects. So kids who have differences in their heart when they're born, or differences in their palate, like cleft lip or palate, club feet. And we also see kids with developmental differences, so autism or intellectual disabilities, any kind of delay. And we try to see the child and decide, is this just an isolated problem, or is this part of a larger underlying diagnosis that may have other associated features that would benefit from knowing about in advance so we can look for them and treat them, and also to look for causes. So why did this happen, and is it something that's going to happen again? So we provide genetic counseling to the families and to the individuals themselves as they grow up, because even though we're a children's hospital, we see kids well into, their late teens and some into early adulthood. And so, all you have to do is pick up the Sunday edition of the New York Times, and you know that it seems everything is genetic. So we're awfully busy here, but we certainly enjoy it. And again, benefiting from people who travel here from all over the world, for the renowned surgeons, the heart surgeons, the craniofacial surgeons, the general surgeons, etc. So because of that, we get to see a lot of wonderful families and children who have things that are often quite rare.
Now, I've had several guests from the foundation on the show up to this point, and everybody's done a wonderful job in letting everybody know about 22q. But I'm hoping that maybe you can give us a really good, strong, technical explanation, but kind of put into, I guess for lack of a better term, layman's terms for us, so that everybody who's listening can relay it to everybody else. We've requested that all of our listeners who are enjoying the show let 22 people know about it, and they've all committed to it. So I'm hoping maybe you can help us by giving us a great explanation for everybody.
Sure thing. Well, in thinking about discussing this on the radio, it's very different from something in person or in a lecture where you can use visuals, because it's talking about chromosomes and genes is really abstract. But if you had a leaf handy, if we were a little later in the season, and you took a leaf and you held it up to a light, you'd actually see that it's made up of boxes. And so all living things are made up of boxes, which are what we call cells. And inside every cell, there's sort of the middle of it, the center, called the nucleus, and inside the nucleus are chromosomes. And chromosomes hold the genes, and the genes determine everything about us. So if you think about a chromosome as being like a train, there are stripes on the chromosome, just like there are boxcars on a train. And if you go to www.22q.org, you'll find this explanation along with some visuals, because this is something that we use as an analogy all the time. So if the train pulled into the station and there were only 10 boxcars instead of 11, you'd notice. And so when you look at a chromosome under the microscope, if there's a stripe missing or extra, usually we can see it. But the genes are the packages inside. So if there's just a change in a gene, or there's a change in a few genes that they're missing or extra, that's not necessarily something that you would see under the microscope. And human beings are made up of 46 chromosomes, so you have 23 pairs, and they start with the biggest, number 1, so they're numbered, and then they go to the smallest, number 22, and everybody receives one of each chromosome from their mother and one of each chromosome from the father, so you have pairs of each of them. And then if you're a male, you have an X and a Y, and if you're a female, you have 2 X's. And so we know that all of the chromosomes are important and all the genes are important, and there are only so many systems in the body that could be affected. So having this tiny little piece of chromosome 22 missing sets up a blueprint for the body to form a certain way. So just like it will set up a blueprint to have blonde hair and blue eyes, or brown hair and brown eyes, or brown skin, or light skin, it determines how the heart forms and how the palate forms and things like that. So a long time ago here in 1982, well before I got to Children's Hospital, Elaine Sakai, who's the head of the clinical department here, and Beverly Emanuel, who's the head of the lab side and now the head of the whole division, they saw a child who had findings that were consistent with what Dr. DeGeorge had described in 1965, and now bears his name as DeGeorge syndrome. And the word syndrome just means collection of findings. And please feel free to interrupt me at any time if I'm going on too long.
Absolutely not. It's wonderful information. Please continue.
Okay. So they saw a child who had a heart difference and a cleft palate and a problem with her intestines, and she also had calcium issues, so low calcium, and she had trouble fighting infection. And they said, this child looks like she has DeGeorge syndrome because Angelo first described children with immune problems, so trouble fighting infection and low calcium. And then later he noted that some of the kids had heart disease, so the heart wasn't formed normally at birth. We call that congenital heart disease. And so they said, well, let's look at her chromosomes because she also has some other things wrong that aren't typical, and we'll see what happens. So it turns out that that child had a piece of 22 missing and a piece of number 10 missing. And so, at the time, there was no literature to go back to say, what does this mean? So they called Dr. DeGeorge and said, have you ever seen any other kids who have DeGeorge syndrome with a chromosome difference? And he had a couple, and the only chromosome that they had in common was this chromosome 22. And so they wrote a paper in 1982 that was sort of a landmark paper saying DeGeorge syndrome is associated with a piece of chromosome 22 missing. Now, I should go back for a minute and tell you that the French first described chromosomes. And so, as I said, they numbered them 1 to 22. And then they noticed that the top half of each chromosome was a little bit shorter in some instances than others, and sometimes it was almost non-existent. So in French, short is petite, so they called the top arm of the chromosome the P arm, which is the abbreviation for petite, and then Q follows P in the alphabet. So when we say 22Q, we're talking about the bottom half of chromosome 22. And then that 11.2 is telling us which stripe that we're talking about, because you may have heard of 22q13.3, or your listeners may have, and that's way down at the bottom of the chromosome. It's nowhere near where we're talking about physically. So those numbers are important in genetics because otherwise you'd be mixing up apples and oranges. So back to 1982. So they saw that little piece missing, and then they said, well, let's go look at other kids with DeGeorge syndrome and see if any of them have a piece of 22 missing. So sure enough, when they looked under the microscope, about 25% of the time they saw a piece of 22 missing. And so this was important because it meant that, yes, for sure, there were kids with DeGeorge syndrome who had it because of a tiny piece of chromosome missing on chromosome 22. But what about the other 75%? Well, the long story short is that they just weren't able to see that that piece missing in those individuals because it was too small. So from 82 to 92, Dr. Emanuel here in the laboratory and Dr. Scambler, the person I mentioned earlier in the UK, both developed a test for the same area on 22 where you could tell that the piece was missing by doing this very special test where you put a light on the DNA and it floated around in a cell, and when it found its match, it lit up. So you would expect to see like two little pink dots on the chromosomes, and then you'd know the piece was missing or not. And that's called FISH. Fluorescence, meaning the fluorescent light. In situ, it was in the cell, and hybridization, if the piece was there, it would stick to it and it would light up. Kind of like, going to a parking lot where you already have an assigned spot, and, you're driving around and you see your spot and you get into it and everything's fine, but if somebody's in your spot, you can't park, so you're in a problem. The same with the piece of 22 missing. Once we had that test, then we could go back to all those kids with DeGeorge syndrome who tested negative when we looked under the microscope, and sure enough, almost all of them had a piece visible under the microscope once we went to this new test. And that was sort of a revolutionary moment in the history of 22q because now we could identify almost all the children who had the piece missing, and it was a better test. What happened next is that we had seen a paper that was written by Dr. Bob Sprinson, who was in New York at the time, and Rosalie Goldberg, who was a genetic counselor who worked with him. And they had said that there was overlap between DeGeorge syndrome and a diagnosis called velocardiofacial syndrome, and I don't know if you saw or read anything about that, but those are kids who had velo, which means palate, so they had palate differences, mostly cleft palate. They had cardiac or heart differences, and then they had some mild facial differences as compared to their parents. And so Dr. Sprinson and Rosalie Goldberg said, well, some of the kids who have velocardiofacial syndrome, or VCSS for short, also have calcium problems. Some have immune problems, and the heart problem's kind of similar to that in DeGeorge syndrome. So we rang up Dr. Sprinson and we said, did you think about testing your patients for 22q? And long story short, most of the kids with velocardiofacial syndrome also had the piece of 22 missing. And concurrent to this, we saw that there was a group of kids described in Japan with something called conotruncal anomaly face syndrome, which is a big word just to mean the same kind of heart difference that was seen in DeGeorge syndrome with some mild facial differences. And if you can believe it, we didn't have computers back in about 1992, so we faxed to Japan and said, maybe you want to think about this, and maybe the children over there have actually have a 22 deletion, and sure enough, they did testing in those children, and those were deleted. So what we realized is that this tiny piece of chromosome material missing in DeGeorge syndrome was really not as uncommon as we thought originally. And that really these were all kids who had the same thing. They had a piece of 22 missing. They just didn't know it because one was diagnosed by an endocrine doctor, the hormone doctor. Another was diagnosed by a speech therapist because of the palate difference, and the people in Japan by a cardiologist because of the heart difference. And it wasn't until we were able to step back and look at this picture with a laboratory test that we were able to say, geez, this is really all the same thing. Not every child has the same symptoms, but they have the same etiology or the same cause for the problem. And I'm going to stop right there for a second and see if you have any questions.
Actually, that's a ton of information. Thank you so much, Donna. One of our listeners.
I have more, but I figure I better stop.
One of our listeners. We have a very diverse listener group, and one of our regular listeners is a young woman named Bailey. She's 20 years old. She's a college student, and she is enamored with you in this, and she's very much into genetics, and she actually sent in some questions and asked me if I would please ask them of you and see if you can answer a couple questions for her. Do you mind doing that?
Not at all.
Okay, so Bailey wanted me to ask you if your research is spread across different projects other than 22q.
So I actually consider myself on the clinical side of things. So I'm a genetic counselor, and I see patients with all kinds of things. So yes, I see children and older individuals, and at times women who are pregnant who have a fetus with an abnormality of some sort, in conjunction with the people who do that next door at the University of Pennsylvania, and also we have a big fetal surgery program here. So absolutely, I see children with other diagnoses, and we have other research projects that we have going here as well. Some of them, many of which have to do with other craniofacial abnormalities. But yes, we have sort of our finger on the pulse of everything in the genetics world that applies to the pediatric population and pregnant women.
She also wanted me to ask you if the award that we spoke about earlier that you won, if that was for a specific project or research that you worked on, or was that just for your life's work attached to 22q?
So, we'd probably have to ask the committee that gave the award, but I think the essence of the award is more a lifetime achievement. And I think when they wrote what you read, essentially, it was to honor individuals who have spent a long time working with families and with children who have the 22q and 1.2 deletion syndrome. And who contribute scientifically, clinically, and then also, as I mentioned, Pete Scambler runs marathons for Max Appeal and raises money for them. I think, that there's some component to this where you really want to help families and you want to help move things along because, you want better treatment and you want better outcomes and whatever you have to do to help with that. So, I've been with the 22Q Foundation from its inception. I was a founding member, and I feel that I was instrumental in bringing 22Q at the zoo to the forefront and supporting that going forward. And so all those great things, we've had 22Q in zoo, we've had Night in Venice, I slice and mean salami, and I like to have fun coming up with some of these names and things like that. So I think it was an honor not just for one singular event, but for a collection.
And her final question, and thank you so much for answering them. She's actually, we have an interactive chat room, and she's very thankful to you for taking the time to answer her questions. She wanted to know why you chose to go into this field.
Well, as an undergrad, I was a biology and sociology major, and actually majors, I guess. And so I knew that I really loved science and I loved medicine, but I wanted to have that social science component to things. And so genetic counseling was really an opportunity to combine those things. And for sure, sitting here now, had I chosen another path, gone to medical school or basic science research, which I worked in a pharmacology lab for 2 years before I went to graduate school. I don't think I would be here now. So, excuse me, a little bit like the Jack Frost poem, The Road Less Taken, I think it's made all the difference.
Donna, maybe you can answer this question for me. What stage of development does the deletion occur, and can it be detected if there's tests that can be done during that period, during that gestation period?
So that's a really great question. The deletion is actually there in either the one egg, the one sperm, or the first cell of development. Normally it's in the egg or the sperm. So at times that's because one of the parents has it. And if a parent has the deletion, then half of their egg or sperm cells have the deletion and the other half don't. So that means that a person with this has a 50% chance of passing it on to their offspring with each pregnancy. And most of the time, though, that's not the case. About 90% of the time, neither parent has it. And it just happens that the piece falls out as a result of the way the chromosome is formed. So what you may not know is that you have very early egg and sperm cells that have to go through a stage where they come together and there's crossing over, and it's sort of how the genes get mixed up, and so that you're not a clone of your parent, so to speak. And then there's a halving stage where, the early egg goes from having 46 chromosomes to only having 23, and the same thing happens in the sperm, and then they come together and you're back with the right number. Along that way, when the chromosome lines up to exchange material, that piece sometimes just falls out because the chromosome thinks it's at the right place for that crossover, but it's really not, because there are areas above and below the 22q and 1.2 region that look very similar, and so it's just a mistake in this crossing over process. So it happens, and then it's in every cell of the body, because once that first cell is there with the deletion, it's like a Xerox machine. It just makes one after another after another. One cell goes to 2, 2 go to 4, 4 go to 8, 8 go to 16. Now, whether or not you can detect it, you can, but you have to know to look for it. So if one of the parents has it, and they say we want to have prenatal diagnosis, then certainly there's a test that could look for it. If they say we don't want to have a pregnancy and have it tested, but we want to avoid that 50-50 chance, just like you could do for people with infertility, with in vitro fertilization, you could check the embryos and implant the embryos that don't have the deletion using in vitro fertilization. But in a pregnancy where there's no history, unless there's a sign on ultrasound, so unless they see that the heart is formed differently, or that there's a cleft palate, or that there's too much fluid or club feet or things like that, there's no reason to test the pregnancy. And that's why some individuals, I know you heard from Sheila Camden last evening, she probably talked about Aiden's Odyssey. There are some kids who come to attention right out of the gate because they have a heart difference that's, the deletion is found in 50% of kids with that specific heart difference. But then there are kids who don't have those very typical symptoms, and they can go for years and sometimes even decades without being diagnosed. And we have adults who never knew that they had the deletion until they had an affected child, and then, everything sort of came together.
Donna, do you have is there anything, thank you again for being on and just sharing so much wonderful information. That's unbelievable, the wealth of knowledge that you have on this. Is there anything that you've written or any articles anywhere that anybody can go and find to kind of keep on hand for a refresher on any of this?
Sure. Well, I think in terms of the lay information, if you just go to www.22q.org, pretty much it's all there. I know it's on the Dempster Family Foundation website as well. It's on Max Appeal's website. If there are articles of interest, I write something for gene clinics or gene tests, and that's a place mostly for professionals to go and read about, a little more detail in terms of how this happened and what are the symptoms associated and whatnot. That's not in lay language, and that's in the process of being updated right now, but it's pretty darn up to date. Ann Bassett, who's the medical director for the 22Q Foundation, is a psychiatrist in Toronto, and she and I and a group of people from around the world put together healthcare guidelines, management guidelines, that were published last year in the Journal of Pediatrics. And for anybody out there who wants sort of a checklist of what to think about, what needs to be done annually, things like that, that's a great article and a great reference, and something that we worked very hard on. We started in 2006, and we had multiple consensus meetings just to make sure that we got it right, because this is a document that isn't just used in the U.S., but it's used in the U.K. and in Europe, and so it had to transcend in Canada and had to transcend healthcare systems and things like that. So it's a very broad brush, but it's also something that's quite good. I mean, the other thing is, because we started seeing patients back in 1992, just because we developed one of those tests that I mentioned, we became a center of excellence for this, a clinical center of excellence. So we have patients that come from all over. We've seen well over 1,000 children. So we started a newsletter that we have for families. So, if anybody wanted any of these things or any articles, they can always contact me by email, and I'm happy to put you on our mailing list and send you whatever we have. And some people need articles about feeding or about orthopedic issues. We have all those things, and we're always happy to mail them to families. So, just give us a holler. Probably the best way to reach me is by email, genetics@email.chop.edu, or mcginn@email.chop.edu. Chop decided that McDonald McGinn was too long. Or I can be reached by phone, 215-590-2920.
Thank you so much, Donna. One final question on a more personal level that Bailey from Oregon had sent in, and I told her that I would ask this, and she wanted me to tell you thank you very much for spending a couple of time to address her questions. She just wanted me to ask you if you had any personal advice for someone like her who's looking to get into a similar field.
Well, I can tell her that I'm happy to talk to her by phone or by email. If she's from Oregon, I know people there because we had a fellow who went there and is now head of clinical genetics out there. There's actually a pediatrician who came to the meeting in Florida, a big international meeting in Florida, who's setting up a 22Q clinic out there. I know a bunch of genetic counselors, and so I'm happy to talk to her about the educational requirements and also about how, she should probably go and spend some time with somebody and shadow and see if she likes it. I have a senior in college and a high school student, and I always say to them, you have to really love what you do, because you spend more time doing that than probably anything else, and I'm sure that's true for you, Joe. You probably absolutely love being on this radio. And so, you never want to watch the clock. You really want to enjoy your work. And the other part is, if you can give back, then it makes it all worthwhile. So if you can combine what you love and giving back and making a difference in the lives of a child or a family, then it's phenomenal. Phenomenal. And by the way, I'm not in any hurry to go up, so don't feel like, you have to let me go. I'm happy to answer any more questions. I'm happy to talk about the diagnosis. I'm happy to talk about newborn screening. We have a big push for newborn screening. Whatever you need, I'm happy to do.
Yeah, I mean, the floor is yours. I don't have any more specific questions. You've more than covered all the questions I had, and I think that you've just done a remarkable job in educating our audience. But, I mean, the floor is yours. If there's anything else that you want to express, feel free.
Well, the only reason I'm still talking is because I think if there are families listening, there are a few things that are confusing. One of them is, why does one child have everything wrong with them and another child have very little? Or why does that parent come to attention only after their child is diagnosed? And, originally when we started studying and thinking about kids with 22q, we thought, well, this is easy. If you have a big deletion and a lot of genes missing, then you have the most trouble. And if you have a small deletion, you have the, little trouble. And obviously, if that were the case, we'd be done. We'd have this all solved by now. But we were naive and we didn't realize how complicated it was, because almost all the kids have the same size deletion with about 40 to 50 genes missing. And we're just now understanding what some of those genes do. And one of them, for instance, TBX1, contributes to the way the heart is formed. And there are others that we're learning about, but we still don't know, what they all do. And we're also learning that, you have genes on all those other chromosomes. So there are things called modifier genes. And so maybe you have the deletion, but you have a gene on another chromosome that protects you from having congenital heart disease, or that pushes things over the edge and causes the problem to be there. So we're doing a lot of work now, and we have a big international consortium where everybody's cooperating and collaborating to understand why those things happen. So that's one thing. The other thing that we're doing, and again, we work really well at this big international level, which we have to do because, you need to combine resources if you're going to make progress. And so one of the other things that there's a push for is newborn screening. And so a group of us that included representatives from the 22q Foundation. Carol Cavanagh was there. Julie Wooten came from Max Appeal in the UK. Sheila Camber was there from 22q. Michelle Breedlove-Fells was there from the Dempsters. And then Allie Mitchell and Jack Rudis from Children's in Milwaukee, because they developed this very neat newborn screening test. And Ann Bassett from Toronto, and I was there from Philadelphia. And we said to the federal government, listen, this is really common. And if we can identify kids early, we can make a big difference. Not only in helping to save the lives of children who may not have been detected with congenital heart disease, because, the test takes about 6 days to come back, and it may be too late for some of those kids. But we know, for instance, and again, I know you heard from Carol Cavanagh earlier, her son Lewis had a calcium problem, and he was having seizures related to that, and they didn't know it. And, every time you have a seizure, you theoretically lose some neurons. And what does that do to long-term outcomes? Well, we don't want to wait and see. We want to identify every single newborn in America and on this planet who has 22q, so that we make sure that they get the best care as early as possible. And so, if anybody out there has any connections with anybody who can help support newborn screening, we would appreciate it, because we will go back to this Secretary's Advisory Committee on Heritable Disorders and Newborn Screening to ask again to add 22q, because the test is there. They just need us to do some pilot studies, which everything, always revolves around money. But eventually the money will be there, the data will be there, and we'll be able to go back to them. And, we need the public support for that. The other thing I would add is that we need the public support and family support for these studies on modifier genes. We're also working diligently right now to put a brain behavior consortium together. Not sure, how much did you hear about the brain and behavior so far on your show?
Wow, I'm overwhelmed with information, Donna. Just a couple questions that I'm curious about. What's the average age of a child who's diagnosed with 22q?
I don't know if we have that information, and I think it depends on where you're seeing patients. So here in Philadelphia, it's much younger because we see children with congenital heart disease flown in here from all over the place, including the military who were stationed in Germany, et cetera. So we see a lot of young kids. Not that we don't diagnose older individuals as well, but for instance, in Dr. Bassett's clinic in Canada, she's seeing adults. And so the ascertainment bias really sort of clouds that answer. So until we do newborn screening and we know how many people there are out there, and until people know about the diagnosis and start testing for it, I don't think that information is going to come quickly. But what I wanted to mention to you when I talked about the brain and behavior, I don't know if you talked about some of the brain and behavior differences that kids with this have in terms of ADHD and autism and anxiety, and there's a percentage of individuals who have psychosis as well. One of the other things that we're desperately trying to pull together is more work in that area so that we can move towards better treatment. And the National Institute of Mental Health actually just put out a call for a collaborative project to study that. And so we're all working very hard to try and pull that together so that, when the next child comes around who may need help in that area, we have it completely worked through. We know how to help, and we know what's available to help. So I'm sorry, back to your question.
Are there any noticeable, aside from the noticeable signs, I guess basically what I'm asking you is, if a child doesn't have the noticeable side effects, what are some of the early signs of 22q?
Well, that one clinical name, velocardiofacial syndrome, suggests that there are some facial differences associated with this. I can tell you, and from Julia, that children with the 22q deletion and adults are absolutely gorgeous. And unlike some other diagnoses that are very recognizable even to lay individuals, individuals with 22q are unlikely to be noticed, just sitting in a waiting room. And so that's one of the problems with, diagnosis. And we think that individuals are not coming to attention because they can look and seem so much like everybody else. And so there are some very subtle features that if you see 1,000 children with this, you could list or you could point out. But in the main, it's a very difficult diagnosis for that reason. I think the way that you come to the diagnosis is to say, okay, a child has the very classic features of DiGeorge syndrome. They had a heart problem, a calcium problem, and an immune problem. Okay, that child should be tested for this. But most of the children don't present that way. Many of the children present with terrible, terrible feeding problems. And sometimes that's related to the calcium problem. Sometimes it's related to the fact that 35% of kids with this have really terrible trouble with reflux and with the food just moving through the entire gut and with severe constipation. And when you're constipated, you're not hungry. And when you're constipated, you reflux more. And so those kinds of things. And when they reflux and they vomit, it comes out the nose. And, we'll hear families say, we said to the doctor the milk came through the nose, and they said that's okay. It's really not. It's abnormal for milk to come out of the nose, unless the child has another structural abnormality of the bowel, and it's just so forceful that it comes through. So, those are things that are generally subtle. And, it's hard for pediatricians to see complicated kids. They have a lot of patients, and they have to get through them quickly, and the reimbursement is poor, and so healthcare is complicated. And I think a lot of the kids fall through the cracks just because, not because the pediatrician isn't well-meaning or the family doctor isn't well-meaning, it's complicated. And I think that's one of the reasons why, one of the other things we're trying to do is to come up with guidelines for clinical centers of excellence and then help support them around the country and around the world. There's no reason why we couldn't have what Jerry's kids is to muscular dystrophy, or the cystic fibrosis clinics aren't for kids with CF. We need clinics around the world for kids with 22q, and we need to find a way to fund and support them, and that will make a difference in the care for these patients and in the identification of these individuals. I know I don't let you get a word in edgewise.
No, it's okay, Donna. Thank you so much for everything. We actually have to get going. We have a schedule that we're trying to keep, but I appreciate your time so much, and thank you. I can't thank you enough for coming on and giving us your knowledge for a little while here. And just really quick, is there anything else that you want to let anybody know of before we let you go?
No, just thank you so much for doing this. A shout out to Joe and Paula for inviting you to do it, and to everybody who's participating. We so appreciate it. And to my good friend Ryan Dempster, who I know is up at 2 o'clock.
All right, Donna, thank you so much. We really appreciate it. And please, if anybody wants any more information, Donna gave all of the websites at 22q.org. Check it out. She made a gracious offer to get in touch with her directly for the newsletter information. Reach out to her. She seems like a wonderful person, and I appreciate the time you spent with us. Thank you so much.
Thank you. Bye-bye now.